So now we are going to start talking. So there is some information about plant cell vacuoles. If you read it yourself, you will probably be able to understand much better than what I can teach. As I keep saying, my knowledge of plant is really, really weak. So I'm going to avoid these two slides and go directly now to the last aspect of this lecture that I wanted to, of this topic that I wanted to discuss, and that is the endocytic pathway. So far we have looked at the exocytosis. Exocytosis is the movement of molecules outside the cell. What I want to talk about is the endocytosis now. So for endocytosis, we are talking about the uptake of cell surface receptors, bound extracellular ligands, which can come inside the cell. This has enormous implications for signaling, and we will talk about that for a few moments. We will talk about phagocytosis, which, as we know, is the uptake of particulate matter. So cells are capable, specific cells like dendritic cells or macrophages, they are capable of extensive phagocytosis. They can take in dead cells, they can take in um, bacteria, viruses. All of these can be then taken in by the phagocytes, and they can be degraded. Endocytosis is divided into two categories. Penocytosis. Penocytosis is the non-specific uptake of extracellular fluids. You perhaps have come across this when you were in middle school or even less as cell drinking, right? So this is a very, very colloquial term used for cells, in that penocytosis is called cell drinking because it's taking up extracellular fluids. Obviously cells can't drink, but it's very non-specific, and it does not involve strong regulation so far. But there is another part which we will talk about more, is receptor mediated endocytosis. So this means that specific extracellular ligands that have bound to their receptors will be taken in by endocytosis with the main idea of stopping the reaction. So let's say that we have a receptor, and again, let me try to draw something here. So let's say this is our cell, this is a cancer cell, and then we have a receptor here for epidermal growth factor, EGF. Okay, so epidermal growth factor binds to the EGFR, and when it binds the EGF to the EGFR, this results in mitogenic signaling. So mitogenic signaling basically means that there will be mitosis happening in the cell, and the cell will divide. Now, in a normal cell, not in a cancer cell, but in a normal cell, this EGF, EGFR signaling is very fine-tuned. So the cell should divide only when it needs to divide. At other times, it should not be dividing. In order to control it, one thing that can happen is this particular EGF, EGFR complex can be taken into vesicles inside the cell. So now it will look like this. It will be a vesicle inside the cell with the EGF, EGFR, and this vesicle will then come inside the cell, and it can actually combine with the lysosome, and it can be degraded. So in this case, this EGF, EGFR signaling complex, this whole complex, is brought into the cell with endocytosis, and this vesicle can actually be fused with the lysosome, and the EGF, EGFR complex can be degraded, which will then prevent the signaling from occurring any further. So we will talk about that in a little bit more detail. So receptor-mediated endocytosis, which is also called RME, is carried out with the help of structures called coated pits. So substances that enter the cell through receptor-mediated endocytosis, they become bound to coated pits on the plasma membrane. So I would like you to remember how the plasma membrane looks. And then if you can imagine a lattice of proteins on the membrane, and we'll look at some pictures in a moment, these coated pits are made up of clathrin again. I will remind you at this point that clathrin was found in the trans-Golgi network vesicles that were going towards the lysosome. Clathrin is also found in these coated pits, and they will, the clathrin coated regions then invaginate, they move into the cytoplasm, and then they pinch free of the cytoplasm. So it works somewhat like this. This is the membrane, then we have an invagination, and then we have the pinching off, right? So this is how this whole process works. When viewed from the cytoplasm, so when we are viewing from the cytosolic side of it, so from over here, this is the cytoplasm, then somewhere in this region where the invagination will happen, will have what is known as a coated pit. Okay, so the coated pit is, it appears as a honeycomb. So they are polygons resembling a honeycomb. Honeycomb is, what was the Turkish word for it? We like eating it very much. You know, the where you can get the actual honey. So it resembles the honeycomb, and this geometric design is derived from the specific structure of the clathrin blocks. We'll look at it in a moment. So this is how it looks. So inside the cell, in the cytoplasmic side, so exactly over here that I was trying to draw, there will be a structure formed, which is called the coated pit. And this defines the area where endocytosis will happen. So you can imagine that this coated pit will be formed if we are talking about recycling of EGFR or degradation of EGFR, the epidermal growth factor receptor. Then this can form actually just below where the EGFR is located. The coated pit can be formed, and then this will then invaginate and it will form a vesicle inside the cell. So these are coated pits on the cytosolic side of the membrane. So when we look at how the coated pits are formed, they are formed of clathrin. This clathrin has a different shape from the clathrin that we had in the vesicle that was formed at the trans-Golgi network and going towards the lysosome. It's also clathrin, but remember we have families of proteins. This clathrin has a slightly different shape in that it has this triskelion shape, which basically means that it has 3 arms that are moving outwards. And the triskelion shape of this clathrin allows the clathrin to bind to an adapter. Again, we've come across several adapters so far. GGA was one of the adapters. And in this case, the adapter is called AP2. I don't expect you to remember what these adapters are. I just want you to appreciate that, you know, how the process of endocytosis is happening. So let's look at it. Here we have a membrane that is invaginating, and on the surface of the membrane, this is the coated pit. So we have the clathrin lattice. So basically it forms a layer around this vesicle. Think of it in 3 dimensions like a football, and all around the football we have this lattice of proteins. And so as soon as it is formed, it still has a link to the membrane, right? So if I were to draw it in my own hand here, this is what we are seeing here. In this case, what we have is this. And in the last stage, what we have is a pinching of the membrane where it still is in contact with the plasma membrane. And once it pinches off, we can have the endocytotic vesicle. This pinching off of the membrane in this region in the middle is carried out with the help of a protein called dynamin. Now, dynamin actually uses GTP hydrolysis. So in this case, we are hydrolyzing the GTP. This is not a simple G protein. This is a protein that hydrolyzes the GTP, uses this energy in order to pinch off, in order to squeeze this end of the membrane so that it forms an individual endocytotic vesicle. And you can appreciate now, I'm sure you understand how, so you can see the triskelion here, 1, 2, 3, 1, 2, 3. So this triskelion basically enables this lattice-like structure on the surface of this endocytotic vesicle. Inside it, we have the membrane, and now within the membrane, we have the cargo. GTP hydrolysis is very important for the pinching off of this membrane in order to take the endocytotic vesicle inside. If we use something called a GTP gamma S, this is a very interesting experiment that very often is carried out in laboratories, where if you remember, the 3 phosphates of ATP, GTP, doesn't matter, are called alpha, beta, and gamma, right? So if I draw the ATP molecule like this, just as a cartoon, we have phosphate, phosphate, phosphate. And this is the alpha, this is the beta, and this is the gamma. And this is true for ATP and for GTP. So if this last phosphate, the gamma phosphate, is replaced by a sulfur, so again we have the guanine group, we have phosphate, but now we have a sulfur. So this is now called GTP gamma S. The same thing can be done with ATP gamma S as well. Not this experiment, but if you want to show whether ATP hydrolysis is important for a reaction or not, you will use this ATP gamma S, which cannot be hydrolyzed. GTP gamma S cannot be hydrolyzed and it cannot produce energy because you don't have a phosphate group here, you have a sulfur group, and breakdown of the sulfur group does not result in the generation of energy as the phosphate group does. So if you were to incubate this reaction with GTP gamma S rather than GTP, then you can see that the dynamin basically keeps forming layer around this, the pinching off part, but it is unable to pinch it off. Rather, the membrane keeps growing and growing and growing, as that you can see in this corner over here. The membrane keeps growing, but the dynamin does not have the energy to pinch it off. So you end up with a structure like this that you can see on the right-hand side. This was a very, very cool experiment showing that the breakdown of GTP is actually necessary for the pinching off of these vesicles into the for their release. So the endocytotic pathway, let me talk a little bit about the endocytotic pathway. We'll keep this picture open and I'll try to explain to you when these endocytotic pathways are carried out in the cell. I gave a little bit of a brief explanation for it a few minutes ago. So if we want to bring in a signaling receptor, so these are all receptors that are found on the plasma membrane, and these receptors are capable of binding to their specific ligands, and they will lead to a signaling pathway happening inside the cell. Now, if this signaling pathway needs to be regulated, then one way of regulating it is by bringing the receptor to the inside of the cell. One may keep the receptor inside the cell and then recycle it back on the membrane. So this is done very often with membrane receptor called transferrin. Transferrin is a membrane receptor that is responsible for the movement of iron into the cell. And there are many functions that the iron can carry out within specific cells, but we don't need the iron inside the cell constantly. We can move the iron only when there is enough iron in the environment. So in this case, what happens is the transferrin receptor is endocytosed inside the cell. It is kept inside a vesicle, inside an endocytotic vesicle, and then when it's needed again, it can be recycled back into the membrane. Alternatively, if this is a signaling pathway that needs to be regulated very closely, such as signaling for growth factors, then we can have the growth factor and receptor brought inside the cell with the help of, again, a clathrin-coated pit that you can see here. Now it has formed an endocytotic vesicle, and this endocytotic vesicle can actually then combine with a lysosome, and it can then get degraded. So we can actually stop signaling in the cell by bringing the receptor and the ligand into the cell in the vesicle, and then by combining this vesicle with the lysosome, so that this will then get degraded and signaling will then stop. Another way in which this signaling pathway can work is when we have the lysosomal enzymes. So remember, the mannose phosphate receptor is carrying the lysosomal enzymes to a late endosome, and this late endosome is now going to be converted to a lysosome. When is it converted to a lysosome? It is converted to a lysosome when the pH inside the cell becomes low. So as soon as the VATPases, these are the proteins that are proton pumps, when they start moving the hydrogen ions in, the pH in this structure decreases, and now it's called a lysosome. So one other way in which the recycling can happen is if it's a housekeeping receptor, or if it is a receptor that can be recycled back because signaling is needed, then this receptor will then move back to the plasma membrane. It will fuse, the vesicle will fuse with the plasma membrane, and it will take the receptor back into the membrane. Okay, so either when you are pinching off this vesicle, if there is a housekeeping receptor coming, that can be sent back. The non-housekeeping receptor, which is the signaling receptor, can actually be degraded. So one can actually even select for which receptor gets degraded and which receptor cycles back to the plasma membrane. This is, again, a very, very interesting, very useful pathway, actually, for the cell to regulate signaling activities from receptors that are located on the membrane. So endocytotic pathway is following internalization of the vesicle. The vesicle-bound materials are transported via vesicles and tubules that are known as endosomes. So we always represent these vesicles as circles, but sometimes they also have the shape of tubules. That's something to keep in mind. Oops. Early endosomes are located near the periphery of the cell. So early endosomes would be located near the cell membrane. They are generally called early endosomes because they have a relatively higher pH. Their pH is closer to neutral. Early endosomes will sort the materials and send the bound ligands to late endosomes. So whichever needs to be degraded will be sent to late endosomes. Late endosomes tend to have slightly different composition. They may have more enzymes that are important for degradation. Their pH will be slightly lower. Late endosomes are generally closer to the nucleus, and they may also be called multivesicular bodies. So here we can see a late endosome, multivesicular body, which will then eventually, when the pH becomes even lower, it will become a lysosome. I am not going to be talking about LDL and cholesterol metabolism, simply because we will talk about it a lot in your physiology lecture next year. So I'm going to stop sharing for now, and I will open